Welcome to our new video. We are starting the renal physiology. We are in the first lecture of our renal physiology. So basically, the first lecture we will start with the basic anatomy of kidney. Okay, the physiological anatomy to be more specific. So the anatomy of kidney. So what is the kidney? In the renal physiology, kidney is the most important organ. Okay, so kidney is responsible for excretion. We all know excretion as a in superficial knowledge we know kidney is important for excretion. Okay, so starting about the kidney. Okay, the kidney consists of this structure. Let me just highlight it, this brown highlighted structure. Okay, the outermost covering of kidney. Okay, it is basically the renal capsule. Okay, this is the renal capsule which protects the kidney from shock absorption. Like for example, if a kidney gets a shock, suddenly get a shock. Okay, so the renal capsule basically helps in shock absorption. Okay, now these we know that from the kidney one tube comes. Let me just highlight it, this tube. Okay, so basically this tube contains urine and it goes to the urinary bladder. So what is this tube? Yes, it is the ureter. So let me just write it here. Okay, this is the ureter. Okay, so now coming to this pyramidal structure. Okay, this one, then this one, then this one, then this one, this one, and this one. So what are this pyramidal structure? So basically these are pyramids present in kidney, so they are known as renal pyramids. Okay, so I will just write it here. This is the renal pyramids. Now coming to one of the most important concept. Okay, the major calyx and the minor calyx. What are the calyx? So basically these calyx are the structure which conducts the urine from these pyramids to the collecting ducts of the nephron. We will study about the nephron. The major calyx and the minor calyx basically collects the urine from the collecting duct and it goes to the ureters. Okay, basically conduction of urine. So the calyx, okay, or the pathway which connects the renal pyramids, each renal pyramid is known as the minor calyx. So this is one of the minor calyx. Okay, now let me just highlight some structure. Okay, so basically this now the what is a major calyx? So basically it is a union of two minor calyx, two or more minor calyx. So basically this is one minor calyx and this is one minor calyx. So basically this structure is known as the major calyx. Okay, so this is the major calyx. Now coming to the connection between the major and minor calyx to the ureter. So basically this structure is the renal pelvis, where all the pyramids open via their calyces and then the pelvis conducts it to ureter. Okay, so now we have studied about the two layers of the kidney. Basically we study the renal cortex and the renal medulla. So what is the renal cortex and renal medulla? So basically the structure here, so this I have made the dotted lines, the dotted lines here, the dotted lines here, dotted lines here, dotted lines between the pyramids. So the deeper to renal capsule and the structure which is outwards from the renal pyramids is the renal, it is the, better we will write is renal medulla. Okay, and the structure which is this, okay, in the dotted line or inwards, it is the sorry, this is the renal cortex and this is the renal medulla. I'm so sorry. So basically you can remember renal cortex as C, covering, okay, covering around the medulla. It is the covering around the medulla. So C for covering, C for cortex. So it is the covering around the medulla. Okay. Now, if we enlarge one pyramid, okay, so we will see much nephrons, okay, very much nephrons we can see in that kidney, okay, in that basically the major in the pyramids, okay, we can see many nephrons. So basically if we enlarge one nephron, okay, so if we enlarged one nephron, it will look like this, okay. So no need to worry, you should draw this diagram only, as this diagram is from Guyton and it is a very classical diagram, okay. It shows many structure, okay, many structure which are not like we tend to draw like this. Don't draw this structure, like this is not wrong, but this structure gives us very much more information, okay. So basically this diagram, why it is important, first we will show the flow of urine, okay. So the urine basically is from here, let me show it in yellow, okay. The urine is basically from here, it goes from here, the arrow represents the direction of urine, okay. So it is go from here, it is will go from this tube, then the downwards, then from this side, then from this side, and then finally the collecting ducts, and then it will open, okay, in the renal pyramids, sorry, the renal pelvis, okay. So now basically we will label this diagram, okay. So this capsule like structure, okay, this is the Bowman's capsule, okay, the Bowman's capsule, and this reddish structure, a tuft of capillaries, is basically glomerulus, okay. Let me mention, it is a tuft of capillaries, okay. Now as I will not mention that this is the afferent, this is the efferent, this is understood, okay, afferent and efferent, okay. So basically the blood is going from here and it is going from there, so afferent and efferent, okay. Now basically the parts of the nephron, the basic parts of the nephron, let me just enlarge, okay, yes, the structure which is from the, like the Bowman's capsule, the immediate tubule, it is known as the proximal convoluted tubule, okay. Now the structure which is like this, okay, a loop kind of thing, so this is the loop of Henle. So basically let me just write it, it is the loop of Henle, okay. Now the loop of Henle consists of two parts, the ascending limb, let me just write, it is the descending limb, as it is descending downwards, okay, descending limb, and this is the ascending limb. So ascending limb basically is divided into two parts, let me just write those two parts, okay. This part appears thin, so it is a thin ascending limb, and this part appears thin, so it is the thick ascending limb, okay. So thin ascending limb and thick ascending limb, okay. This structure, okay, this one, let not confuse you, this structure is basically the distal tubule, okay. And you can see this demarcation of line, okay. So this demarcation of line basically This is the renal cortex. Let me use a different color, a bright one. This is the cortex, renal cortex, and there is a renal medulla. Okay. Now, this structure, okay, this is the collecting tubule where the urine is collected. Okay. Now, many collecting tubule like this one, they collect the urine. Okay. And they open into a common duct. Okay, so this is the collecting duct. And this collecting duct, the major collecting duct, basically it opens in the renal pelvis or the calyces kind of thing. Okay, and then the pelvis. Now, what extra structure we can see here? Okay, why I have chosen this diagram from Guyton? Okay, this structure shows the macula densa. So this extra structure, so basically this one. Okay, I will use a bright color. Okay, so no, this one, no, I will use yellow. Okay, so this structure is basically the macula densa. They are specialized cells. Okay, we will study about them. So basically they are specialized cells which alter the concentration and concentration of urine. Okay, the reabsorption of water, if it is needed or not. So basically it is an important one. So let me just highlight it. Okay, the macula densa is seen. Okay, so this diagram holds so much importance. Like, you can easily like show the macula densa in the cut section of the diagram. Like, if you get a question on macula densa, explain what is macula densa. So there's one tubule going in the glomerulus, other tubule is going out of the glomerulus, one tube is passing through it in between, and you draw the cells. But this diagram gives you the feel where are exactly the macula densa. Okay, so therefore this diagram is much more important. Now, talking about the functions of the kidney. Okay, let me just write it. Functions of kidney. So there is one mnemonic. The mnemonic says a wet bed. Remember it, like a kid is urinating in his bed. So a wet bed. So a wet bed. So A W E T B E D. So what does A stands for? So basically the first and foremost function, it is acid and base balance. Okay, we will see the acid and base balance in deep or in greater details further. Okay, then W stands for water balance. Okay, E stands for electrolyte balance. Okay, then T stands for toxic waste management. Then other B stands for blood pressure regulation. Oh, sorry, there are only this one B, just one B in this mnemonic. Blood pressure management. We have two E's. Okay, so the other E, it stands for erythropoiesis. Okay, and D stands for vitamin D synthesis regulation. Vitamin D is also known as, yes, calcitriol. So basically calcitriol synthesis can be regulated in the kidney. So these are the functions of the kidney, a wet bed. Okay, also we know that the descending limb and the ascending limb, the functions of the descending limb and ascending limb, basically I will not show in this diagram. I will go to another diagram, or I will just write it. We know it, like we have studied it in primary classes. So basically what happens, the descending limb. A descending limb is basically, it is permeable to H2O and is impermeable to ions, and the ascending limb is just the opposite. It is permeable to solutes and it is impermeable to water. While in the case of PCT basically, it reabsorbs the solutes, it reabsorbs water, it reabsorbs glucose, it also secretes some other things like the waste products and all. The DCT basically, it do like reabsorption, but if it is required, it does not do reabsorption, like it does not do reabsorption quite often, but if it is required, the DCT performs reabsorption. Okay, so these functions we know that. Then we will directly move on to the glomerular filtration rate. Okay, so let me just write the heading: glomerular filtration rate. Okay, so what is this glomerular filtration rate, or better known as GFR? We will write GFR. Okay, so it is the amount of filtrate, okay, formed per minute. Okay, so basically it is the amount of glomerular filtrate formed per minute. Okay, it is the amount of filtrate or glomerular filtrate formed per minute. Okay, so what is the normal value of 120, like the normal value of GFR? Okay, so basically normal value is on an average, like it is nearly 125 milliliter per minute. Okay, so this is one of the most important value we should know. What does this signify? The GFR is, we do the calculation like 125 ml per minute. So 125, if we like do calculations and calculate it for per day, so it is nearly 180 liter per day. Okay, but we do not urinate 180 liter per day. We urinate only 1 to 1.5 liter per day. So where does this 99% reabsorptions occur? So 99% reabsorption occurs in the like the whole of the glomerular, whole of the urinary apparatus. Okay, so basically we see 125 milliliter per minute, the normal value of GFR. Okay, now GFR actually is an indicative of kidney function. Okay, let me just mention this important point. GFR is indicative of renal function. Now, how does this GFR occur? Okay, so basically, let me just draw one diagram. I will come in a minute. I will draw one diagram. So basically, if we like, this is the diagram. Okay, so basically I will explain you the statement GFR is the indicative of renal function. First I will explain how does GFR occur. Okay, so basically this is the glomerulus. Okay, only the glomerulus, the Bowman's capsule and the tuft of capillaries glomerulus. Okay, so basically if we just magnify this area, okay, and just we will see this in under the microscope. Okay, so basically we will see this kind of structure. Okay, so what is the structure? So basically let me draw some cells also. Okay, so there are three membranes. Okay, three membranes. So what are the three membrane? So these red cells, okay, these red cells, These are the endothelium. Okay, endothelium of blood capillaries. Okay, the endothelium of like this blood capillaries. Then it is, what is this green structure? This is the basement membrane. Okay, this is the basement membrane. Okay, so this is the basement membrane. Now what are these blue structures, the blue cells? So they are the podocyte. They are the podocytes. Okay, so what happens is the blood is flowing like this. Okay, so blood is flowing like this. So what happens is we have to do the filtration. Okay, so what happens is the blood without proteins, they enters like this. Okay, so why there are no proteins? Okay, so the basement membrane is a specialized structure in this case. The basement membrane contains proteoglycans, and you very well know proteoglycans is negatively charged, so it will repel those proteins and the proteins will stay in the blood capillary. Okay, so what happens is the blood which comes from the endothelium, then the basement membrane and the podocytes. The podocytes are basically, I must not draw like this, this is a very schematic diagram, but I should draw a structure like this. Okay, that was one schematic diagram, but to show the real diagram it is like this. Okay, so basically the podocytes are specialized cuboidal cells. Okay, they contains this slit pores. Okay, this slit pores, and this slit pores allows the incoming of blood from the endothelium, then the basement membrane, and then from the podocytes to the urinary tube. Okay, urinary tubules. So basically without proteins, due to that proteoglycans, the blood flows into the urinary tubules. Now if this proteoglycans, let me just write it here, a slight change in this negativity, it will result into proteinuria or albuminuria. Okay, a slight change. Now this was all about the GFR. So why this GFR is indicative of renal function? So basically what happens is, to check the functioning of the kidneys, we do a test known as inulin test. So this inulin structure, okay, it is a polymer of fructose. So basically what happens, this inulin is given to the patient intravenously, and then its activity is calculated as it passes through the urine. Okay, so it doesn't get reabsorbed and it doesn't get secreted by the renal tubules. Therefore it is a very commonly done test, and the GFR can be calculated by this. Okay, so since GFR can be calculated by this inulin test, so GFR is an indicative of renal function. So if the GFR is normal, the urine output or the urinary system, it can be normal. The further complications can be like the tubular complications. But if the GFR is normal, like the GFR is an indicative of renal function. Okay, the statement explains this. Now we will talk about the factors. Okay, the factors which affect GFR. But before this, we will talk about an important concept, the just a second, the filtration coefficient. Okay, filtration coefficient. So what is this filtration coefficient? So filtration coefficient is basically denoted by Kf. It is, let me just write it. It is basically Kf is equal to GFR upon 10. Okay, so how does this 10 comes? From where does it 10 comes? The value 10, where it comes? So basically for this we will study the determinants of GFR. Okay, the determinants of GFR. Now let's imagine a Bowman's capsule. Okay, so I will be drawing a very schematic diagram. So this is one of the Bowman's capsule. Okay, now let me just complete this diagram, then I will come in a bit. Okay, so basically I have shown the afferent arteriole. I will just write AFF, and this is the efferent arteriole. A very schematic diagram. The blood capillary is stretched. Okay, so there is no tuft of capillaries. Okay, it is to show the the determinants of GFR. So what are the determinants of GFR? So tell me one thing, the blood is flowing from the blood capillary. Okay, so what happens is the blood contains fluid. Okay, so the fluid will exert an pressure. Okay, so how will this pressure, what will be the direction of the pressure? It will be towards the Bowman's capsule. Okay, so let it be PG. So PG corresponds to glomerular hydrostatic pressure. Okay, so the pressure due to fluid, the hydrostatic pressure. Okay, now there are also proteins which are present in the blood capillary. Yes, so these proteins basically exerts a opposite pressure. They will hold the water, or they will the pressure will be opposite. Okay, so let it represent pi, or let it be P O. Okay, so it is PO is basically glomerular colloidal osmotic pressure. Okay, so basically the proteins will prevent it. Now the same hydrostatic pressure will be present in the Bowman's capsule. Okay, so basically the hydrostatic pressure will be towards the capillary. Okay, so basically it represent pi g. Okay, pi g. So pi g represent glomerular, it is the Bowman's capsule pressure. Okay, now there will be no Bowman's colloidal osmotic pressure. Why? Because proteins are absent. Okay, so now tell me one thing, the net pressure. So what will be the net pressure? So let me just calculate it. I will let you know the value. So net pressure is equal to, it will be the PG, glomerular hydrostatic pressure, minus, or rather say plus, pi O, the Bowman's capsule colloidal osmotic pressure. Okay, so it will be like this, but since proteins are not present, the value will be 0. Then minus the glomerular colloidal osmotic pressure, and in the bracket plus the Bowman's capsule pressure. Okay, so basically the value of the glomerular hydrostatic pressure is 60. The value of Bowman's capsule colloidal osmotic pressure is 0 as the proteins are absent in the Bowman's capsule. Then the value of the glomerular colloidal osmotic pressure is basically 32, and the value of Bowman's capsule pressure is basically 18. Okay, and the answer comes to 60 minus 50, we get 10. So the net filtration pressure, okay, so I better write net filtration pressure. Okay, so the net filtration pressure is 10. So this 10 comes from the net filtration pressure. So tell me one thing, the GFR will be some constant multiply this net filtration pressure. Okay, so that some constant is basically the filtration coefficient, and the filtration coefficient formula is GFR by 10. So the filtration pressure is basically 125 by 10, which gives a value of 12.5. Okay, now the determinants of GFR are basically this pressures. Okay, so it is PG, it is PO, and it is pi G. Okay, these are the determinants of GFR. Now we will see an overall factors affecting GFR. Okay, the factors affecting the glomerular filtration rate. Okay, the GFR. Now remember how to write the factors. So I personally write the factors like this. So I first write the filtration coefficient. Okay, so the filtration coefficient KF. So tell me one thing, if the net filtration pressure increases, so basically it comes like it goes till 11 or 12, so filtration is increasing, and since filtration is increasing, what will be the GFR? Yes, the GFR will increase. Okay, so KF increases, GFR also increases, and KF decreases, the GFR decreases. So now the KF, okay, the KF can be like decreased in cases of the thickness of capillary membrane. So if the capillary is too thick, the net filtration pressure can be less, so the GFR drops automatically. Now if also another reason, if there are the functional capillaries, like the functional areas of the capillaries are less, the filtration pressure is less, and therefore the GFR drops. Okay, then after KF I write the three pressures. Okay, so first the hydrostatic pressure, the glomerular hydrostatic pressure. So tell me one thing, the glomerular hydrostatic pressure was directed from the blood capillaries to the Bowman's capsule. So the logic is PG increases, then GFR will increase, and PG decreases, the GFR will also decrease. Okay, so glomerular capillary hydrostatic pressure, if it increases, the GFR increases. Okay, so the PG can be increased like if there is increased renal blood flow. Okay, or afferent arteriole is dilated, so more blood is entering, so the PG increases and then the GFR increases. Okay, then I write the second pressure. Okay, the second pressure basically I write it as the pi G. Okay, the glomerular capillary or the Bowman's capsule hydrostatic pressure. Okay, so basically tell me one thing, it was directed from the Bowman's capsule to the capillary, so if pi G increases, the GFR Yes, this automatically decreases. And if the pi g decreases, the GFR further increases. Okay. Now, the third pressure, it was the glomerular capillary colloidal osmotic pressure. Okay, so tell me one thing, it was directed away from the Bowman's capsule. So if the glomerular capillary oncotic pressure or the glomerular capillary colloidal osmotic pressure, if it is increases, the GFR will automatically decrease, and if it is the P naught or the capillary oncotic pressure decreases, the GFR increases. Okay, so we have completed the three pressures. First we have written about the KF. We have completed the three pressures, so four factors are done. The fifth factor is permeability. Permeability of membrane. Okay, so we have seen the membrane, the three layers. Okay, so if permeability of the membrane increases, so what will happen? The GFR will, yes, increase. Okay, so we have done with five factors, and the last factor is the sympathetic stimulation. Okay, sympathetic stimulation, this is one of the most important stimulation or the important factor which affects GFR. Okay, you should write this as the most important factor. Okay, so if there is stimulation of sympathetic nerves, okay, so what happens? Constriction of the afferent and efferent arteriole occurs. Okay, so since the strong sympathetic stimulation constricts both the arterioles, okay, there will be no effect in GFR. Why? So sympathetic stimulation it will cause constriction of both arterioles, but there will be no GFR change. Why? The concept is auto regulation. Okay, the GFR gets auto regulated, like the renal blood flow get auto regulated, so the GFR remains normal. Okay, so this was all about the glomerular filtration rate. Okay, so now we are going to start about two main processes. Okay, the first process is I will just write tubuloglomerular feedback. Okay, and the other process is JGA, juxtaglomerular apparatus. First we will discuss about the tubuloglomerular feedback, and the juxtaglomerular apparatus will be discussed later. So basically I means you will find me dumb, but like I got confused many times between these two processes, the juxtaglomerular apparatus and the tubuloglomerular feedback. So in the first figure itself I told you to mention the macula densa, okay, in the nephron structure. So you will know the importance of it now. So basically the tubuloglomerular feedback is related to delivery of Na plus and Cl minus. Okay, so what happens is, let me just draw a picture. Okay, I will come in a minute. Okay, so I am back with this picture. Okay, so what I have drawn, so basically we have seen this figure. Okay, so I will just zoom in it a bit. So I have drawn basically this structure. Okay, so what I have shown is like I have shown the glomerulus. Just a second. The glomerulus. Okay, then the afferent arteriole. Okay, the efferent arteriole, and I have shown this tube. Okay, the macula densa. Okay, so in cut section basically we will see like this. So basically this part is the macula densa, or the urinary tube containing the macula densa. This is the afferent arteriole, and this is the, just a second, this is the efferent arteriole. Okay, now to label things, I will just label it and I will then explain the tubuloglomerular feedback. So basically afferent, efferent, arteriole. Then you can see this endothelium. Okay, this is the endothelium of capillaries, and this brown cells. Okay, what are these brown cells? These brown cells. So basically these are modified smooth muscle cells around the blood capillary. So modified smooth muscle cells. Okay, and here's the catch. Listen very carefully. This modified smooth muscle cells are basically the juxtaglomerular cells. Okay, so remember one thing. The juxtaglomerular cells are not urinary cells or the blood cells. They are basically modified smooth muscle cells. Okay, and this tube, the urinary tube, which consists of these cells, they are the macula densa cells. Okay, don't confuse macula densa cells. Now I will tell you the functions of both the cells. Okay, first we will discuss the tubuloglomerular feedback. Tubuloglomerular feedback, we have already written that it is basically the delivery of Na plus and Cl minus to the cells. So what happens if there is decreased blood pressure? Okay, so if decreased blood pressure, there will be decreased renal blood flow. Okay, so renal blood flow will be decreased. Since renal blood flow will be decreased, the decrease in blood flow to macula densa cells. Then if there is a decrease in blood flow to macula densa cells, so for decrease in delivery of Na plus to macula densa cells. Okay, so macula densa senses decrease in Na plus. Okay, so they basically the senses a decrease in Na plus. Now what happens? There will be dilation of afferent arteriole. Okay, and since dilation of afferent arteriole, there will be renal blood flow and GFR back to normal. So if they are back to normal, the Na plus and Cl minus will increase to the macula densa cells. And the opposite is also true. Increase in renal blood flow or increase in blood pressure, increase in renal blood flow, it will increase the GFR. Okay, it will increase the Na plus and Cl minus delivery to the macula densa cell. They will sense that and they will constrict the afferent arteriole, and they will regulate, like they will regulate the renal blood flow back to normal and GFR also back to normal. Okay, one statement I would like to add in the middle, sorry for that. So basically one statement is decrease GFR. Okay, so now this is true, the tubuloglomerular feedback. This is the tubuloglomerular feedback. And remember this diagram, I will give a box for this diagram. So basically this diagram is the JGA complex. The JGA complex also includes the macular densa cells. Okay, so this was all about the tubuloglomerular feedback. Now, what about the juxtaglomerular cells or juxtaglomerular complex? Juxtaglomerular complex, basically JGA complex. The JGA complex consists of this macular densa cells too. So what happens is, if there is decrease in blood pressure, surely there will be decrease in renal blood flow. Okay, the smooth muscles, the modified smooth muscle, which are the JGA cells, the JG cells, they senses it. They release renin. Okay, this renin will go to liver. Okay, this renin will go to liver, and mind it, this is RENIN. The REN is basically the renin which is present in milk protein. This RE∑N is secreted by JG cells. Okay, so the JG cell senses it, releases renin, it goes to liver, and here one procedure occur. This is angiotensinogen to angiotensin 1 with the help of this renin. Okay, now this angiotensin 1 will go to lungs. Okay, and in the lungs one procedure occurs. This angiotensin 1 is converted to angiotensin 2. Basically this angiotensin 2 is a powerful vasoconstrictor, and the enzyme is angiotensin converting enzyme. Okay, the ACE enzyme. So the angiotensin 2 is basically, it is a powerful vasoconstrictor. Okay, so this powerful vasoconstrictor, basically it, let me just write it, constriction of efferent arteriole. Okay, so basically constriction of the efferent arteriole, and then GFR is bring back to normal. Okay, so this was about the JGA complex. Okay, so this flowchart is important in JGA complex. Okay, just like this. Okay, so this flowchart is very important in JGA complex. Also, when there is a stimulation of the juxtaglomerular apparatus, mind it is apparatus, it is not like a single thing. So the JG complex will consist of the JG cells as well as the macular densa cells. So both will act simultaneously. So these processes will occur simultaneously. So this was all about the two important procedure, the juxtaglomerular complex. Okay, the juxtaglomerular complex consists of the macular densa and the JG cells, and how they regulate the GFR. This was an important concept. So that's it for today's video, and other parts will be continued further. Thank you.